I'm very pleased to introduce Marcel Jaspars to you. Marcel's working just up the road in the University of Aberdeen in chemistry, where he's Professor of Chemistry, and he's going to tell us about marine biodiscovery. And I think you're probably the leading marine biodiscoverer in the UK.
Thank you very much, Christine. Thanks for the invitation. My presentation is in two halves, really. The first one is just describing the subject of marine biodiscovery or biotechnology or bioprospecting, whichever way you want to describe it. The second half is more specifically about the work that we do and how that fits into the sort of big picture of things. Just to let you know, I used to give presentations like this as part of the Knowledge Transfer Network, where they still had a section called Biofinancial Business, and I was the blue biotech representative. So I've been to meetings like this before in various places like CFAS around the country. So I tried to make it general. So for those of you who don't know the word marine biodiscovery, it's a made-up word. It just means looking at nature for the discovery of new products and processes, essentially. And so it always starts off with some kind of sampling. It could be marine, it could be terrestrial. And then getting your bioresources. Those could be whole organisms like sponges, fish, whatever you're interested in, or it could be, in our case, quite often bacteria, cyanobacteria, algae, all these kind of other organisms that you might want to look at. That leads to discovery of new materials. And those materials, you can do functional testing of some type. That could be, in my case, a biological testing for discovery of anti-cancer compounds or antibacterial compounds, but also for discovery of new processes, such as finding new enzymes that can carry out new conversions in industry. And then there's a long path to commercialization. Quite often it's shorter for things like enzymes, very long for processes like drug discovery, up to 20 years or more. The reason we use the word biodiscovery and not bioprospecting is the word bioprospecting has connotations of you going in to mine the resource later on. And in most of the cases, we use the idea only. We don't use the actual natural resource to harvest the final product. That is the case in some places. If you take a seaweed or you take a fish and you use that for a particular product. But most of the concepts I'll explain will be for the discovery of the idea and then the production of the material by some other means. Why marine environments? Again, huge diversity of environments, anywhere from the hot vents to the subduction zones, deep trenches, cold seeps, reefs, etc. We're interested predominantly, I started my life off as working on sponges from coral reefs in Indonesia, then moved on to sea squirts in the Pacific, and now much more working on deep sea bacteria and also cold adapted bacteria in particular. I don't know if any of you have looked at this. This is a recent resource, and Christine and I were both on the working group that produced this, and it's worth looking at the Future Ocean Resources document. It describes both deep seabed mining as well as marine genetic resources from areas beyond natural jurisdiction. So it's worth having a look at that. It has this wonderful mapping tool as well as a report that's intended to read for ministers and their researchers, so it's not very high level, but nevertheless it introduced the topic in a very positive way, so I recommend you have a look at that. There's another image there, I think, of the deep oceans. So again, it goes through that. I guess you're all aware of OBIT's tool as well for mapping biodiversity around the planet and the oceans as well. Again, that's a wonderful tool. This is slightly different because it deals with particular environments, whereas OBIT deals with particular phyla. I guess you're all well aware of this is the marine diversity of organisms. This is an old chart I got from my colleague Chris Battershill at Ames in Australia, now at NIWA, no longer NIWA, University of Waikato, New Zealand. And it really just describes in a sort of estimate on the side here, in terms of this logarithmic estimate here, so 10, 100, 1,000, etc., number of species in particular phyla of animal life. You see that the marine animal diversity is much, much greater than that for terrestrial organisms, that there's only one terrestrial-only phylum, whereas there's about 20 marine-only phyla, which means that the marine diversity is huge. And if we start to look at the chemistry of all of those, it's really very diverse and very different from that found on land. Microbial diversity is even greater. This is Mike Ward's from Newcastle's taxose space. So it's a three-dimensional PCA of particular parameters associated with microbial diversity. And what you can see is there's a huge amount of microbial diversity. The marine taxa seem to be far away from anything terrestrial. That's quite interesting to see. So if you look at proper marine adapted organisms, they're very, very diverse. And I don't think we still have any real idea of marine microbial diversity or its true value. So again, we need to really start working on that a lot more. The concept of what we do is really to take marine bioresources and turn them into useful products and processes. My own interest is predominantly in looking for new medicines and molecular research tools, and then enzymes as well. So I won't talk about the enzyme work at all, just talk about the discovery of drugs. On the other side, of course, people are always trying to make personal care products, nutritional products. That's very popular nowadays. It's because the backstory sells the product in many cases, even if it has a better function, better application. And the last application would be things like biomaterials, bone replacements, and things like that. So why would you want to use them? Well, the answer is really that in order for anything to be interesting, it has to have a superior performance to a terrestrially comparable product, or it has to have better economics, it has to be cheaper, essentially, for the same performance. But quite often we look for products that have wholly new functions, wholly new properties. So you might have an enzyme that can do a chemical reaction that nothing else can. And I can point to several examples of that. You might look at small molecules that have drug-like activity with new mechanisms of action that we haven't seen in the terrestrial environment. You might have materials that have new properties, and a great example is a product called gecko, which is a product derived from the gecko. The gecko's food mixed with adhesive threads from mussels and is a reversible underwater adhesive. It's a fantastic idea for a product. And again, it's this kind of thing where you have unprecedented activity that really the subject comes into its own. So some examples of non-pharma products on the market. I would like to go there first. That's more diverse. This is Vent polymerase, which is a high-fidelity DNA amplification discovered from a vent bacterium in Naples. It's off the coast, apparently. Produced recombinantly, again, so not produced from the original organism, of course. This one is harder to find facts about. It's something called Fuelzyme, which is produced by the Beryllium Corporation in San Diego from a deep-sea vent bacterium, we think. Location unknown, you can't find it in the patent. It's an enzyme that might be produced to turn cellulose into biofuels. And again, it's been bought by BASF now, so it's quite an interesting story. But again, we don't know where it came from. It's hard to find this information sometimes. This is a particularly interesting product for screening infrared rays and therefore reducing wrinkles. It's a face cream produced by a company called Sederma, produced from a deep-sea bacterium, again, location unknown. I asked them how much money they were making off it, and Coda said, we can't tell you, but cosmetic industry is worth so many billions per year for the UK. So it's very hard to find facts out about some of these very proprietary products. The last one is an antibiofilm inhibitor from a red seaweed. It's used initially by University of New South Wales research group, Schillerberg and Steinberg, now bought by Unilever for things like medical products such as the catheters and things like contact lenses to stop biofilm formation on them and make better for the patient and better for the doctors as well. In terms of drug molecules, we have, I think, about 9 products on the market right now. The first one to go on the market was the top right one, which is Prialt for chronic pain. So this is the kind of pain caused by nerve damage or limb loss, cancer, virus, often affect the nerves, the nerve endings, and cause the kind of pain that can't be treated with morphine. The cone snail, the magician's or the socialist cone snail, Conus magus, was taken from the Philippines. Group at Utah, Toro Oliveira's group, developed it into a painkiller that had to be injected up the spine. But for this kind of pain, it's the only type of painkiller that will touch that pain compared to morphine, for instance. The second one to go on the market was this one here. It's Yondelis, produced by the Pharmamark company of Spain. It's a very small company, maybe 300 people, that has the biggest pipeline of anti-cancer drugs bar one major pharma company. So it's pretty amazing. We only work on marine species. In this case, the Caribbean sea squid, Echinacea turbinata, was used to produce this molecule, which is now produced in a semi-synthetic process. That means they take a bacterium, they grow it, and they use chemical steps to convert it into the drug molecule. The next one, I think, is this one here, which is called Halaven, which is produced synthetically based on a sponge molecule bottle. So initially discovered in 1986 by Bersuka Uemura in Japan. It's now made totally synthetically as a derivative. It's the world's most complex drug molecule. This one here came originally from a Mauritian sea hare, Dolabella auricularia. Turned out it wasn't from the sea snail at all. It was from the cyanobacteria growing on its back. They slaughtered 2,000 of these poor animals to get 10 milligrams of material, out of which they then developed the drug molecule, which is made synthetically, attached to an antibody, and then used to attack cancers, particularly by targeting those cancers. There's 2 others. This is a very old one. The link to the marine environment is actually quite weak. It's Tethia crypta, discovered in the 1950s. The molecule then leads to the anti-nutrients such as RNA and RSC, which are used for cancer treatment. And this one, last one here, is highly purified fish oil, essentially. Polyunsaturated fatty acids used for patients who have had heart attacks, and then to prevent the second one, they take this very high-strength polyunsaturated fatty acids. So let's see the example of the ICI compound called Halaven. So originally discovered, as I said, in 1986 through bioprospecting. This is Hylochondria opidae, the south of Japan. In 1986, the structure was reported by a group that normally works on marine toxins. That is that big field. The molecule looks like a marine toxin. It looks like oxalic acid, but not quite. So this is the full-size molecule. 1996, it was synthesized in a huge tour de force synthesis. In 2001, they made 200 different derivatives, out of which one was taken forward into clinical trials over the next 9 years. It was approved in 2010 by the US FDA. Current sales are about 350 million per annum. It's not a blockbuster. Blockbuster is defined as 1 billion per annum. But again, you can see it took 24 years to get to the market. It's not a trivial process. that was driven really by ICIDE decided this was a molecule they wanted to work with. It had a new mechanism of action, and they wanted to make it as simple as possible, despite which it is still incredibly complex. So we see that these compounds that we're looking at, they keep being increased. Is linear. It doesn't seem to be exponential or anything. It's hard work to discover new molecules. It's hard work to discover new ones with new activities. So quite often you have either or. You have something that has new activity, it's a known molecule, or you have a new molecule with no activity. So quite often to match the two up. If you have both, and you're very lucky, you're going to show some good publication, perhaps it happened as well. So what's happening so far? So at the moment, we have about 250 molecules from the marine environment in the preclinical pipeline, out of which 15 are in phase 1. The phase 1 trial is particularly the first-in-man trial. Second one is dose escalation trial, where you try and see if it works in patients with only that disease, and if you increase the dose, does it have an increased effect? Phase 3 is the last stage, multi-center, huge international trials. At the end, we have in the clinic about 7 at the moment, drug molecules. Most of those came from the reef environments. There's very few from the open ocean still. Most anti-cancer, because the whole process has been skewed by the National Cancer Institute of the USA, who funded a huge amount of this research in the US over time, since the 1970s. So we see a lot of these drugs coming forward, but mainly for cancer. A few for pain and a few for viral diseases. Most of them have been taken on by small companies, not big companies. The big companies are scared of this kind of stuff. But if any of you are interested in looking at that further, look at the Marine Pharmacology website, which has up-to-date information on drug molecules that are coming through the pipeline. So as part of the process, I've got involved recently through the big European project I was leading called Pharmacy in legal aspects of this, talking about the area beyond national jurisdiction. So the high seas essentially is the area beyond the exclusive economic zone, pretty much beyond 200 miles. It is everybody's, okay? And it's the seafloor is called the area, depending on the continental shelf. It's either 200 miles or 350 nautical miles. It's a 150-mile zone that's under question. But the point is that in that area, at the moment, I could go and collect specimens and carry out scientific research without any intervention. I just need to tell somebody I'm going to do it, and that's pretty much it. So if I was going to collect within a territorial zone, the EEZ, then it belongs to the country of origin, of course. And typically when I've done that in Fiji, I go through 3 levels of permission: the national level, the regional level, and the village level. But I do collect them with prior informed consent. I get permission to do so. I do it on the mutually agreed terms. What is the output going to be? And quite often it's something like, shared research goals, shared publications, and if there's any royalties forthcoming, then we share those. And that's the equitable sharing of benefits. But most of it, for an academic like myself, it's about training scientists in the lab. So it turns out that not all countries have ratified or have mechanisms to deal with this at all. So it's very difficult. There's not very much knowledge among scientists either what they have to do. And we've been dealing with a number of people at the EIS in London to try and make sure that we're doing the right thing at the right time. So our university agreements are now Nagoya compliant. We send them out to partners in other parts of the world to make sure that we have the right permissions to deal with this. So beyond the exclusive economic zone, again, about 60% of the oceans are outside of the exclusive economic zones. Then the United Nations Convention on Laws of the Sea applies. And the point is that actually, the main statement is that it's for peaceful uses of the seas and oceans, the equitable and efficient utilization of resources, conservation of the living resources, and study, protection, and preservation of the marine environment. It's a very lofty goal. I've read the whole thing. It's very broad and general. Some things seem to apply to me. But there's no mention of something which we would call marine genetic resources, because at the time that the convention was framed, the only thing that they thought was mineral resources on the seafloor were important. So they defined resources as the non-living mineral resources, which is interesting. Because in 1982, we were just beginning to find that there were these ocean smoking, the hot smokers or the hot vents, and that there was indeed a lot of life on the deep sea floor. The problem is that there are 2 conflicting elements of this convention. The first one is the freedom of the high seas. You have free passage, you can go fish wherever you want to. And it means that if you go do marine scientific research in the oceans, in the areas beyond national jurisdiction, you can do that freely as long as you share the results. And for scientists, that means publication. And to date, that means that something like the National Oceanographic Centre in Southampton carries out its research, and the information is online. I can go online and find exactly where they've been, where the samples are, etc. On the other side, there's this principle of the common heritage of mankind, that no state shall claim sovereignty over any part of the area or its resources, that means I can't patent something that I find and I can't commercialize it. That's what I take it to mean. There seems to be a conflict between the two. I can't really reconcile those easily. Either I could do what I want and just publish it, or I could do something, but I have to then share all the outcomes. So that's what the problem is. So at the moment, there's a process. In 1982, the convention excluded marine genetic resources, or living things basically on the seafloor. In 1996, it took that long for somebody to point out there was a problem here. It's an article called The Deepest of Ironies, and it's been revised since then. In the 2000s, they started having this ad hoc open-ended working group, and whenever you see in the UN an open-ended working group, it means it's going to take a very long time for anything to happen. And they were, what does it say? To study issues relating to the conservation and sustainable use of marine biological diversity beyond areas of natural jurisdiction. It lasted for 14 years, roughly, before they came up with something. If you read the reports that were happening at the beginning of the 2000s, they weren't very productive. There was people not wanting to do anything to do and block each other's oceans. In 2015, as our project was underway, there was a decision to develop an internationally legally binding instrument under UNCLOS. That meant that they wanted to do something. They had 2 years, 2 times 2 weeks every year of discussion on what to happen. And at the end of it, what came out of it was a document which is essentially the headings of all the things they would discuss in an intergovernmental conference. What they had then is that they called this the elements of a draft text, not the text itself, but what the text should contain in terms of which elements of it should contain. I was quite dumbfounded when I saw the final document after 8 weeks of discussion with 300 people involved. They had got to 13 pages of headings and no changes to anything on marine genetic resources, just the old statements that they started out with because they couldn't agree. Hopefully this year, in September, they will start the Intergovernmental Conference. They'll have a preparatory meeting on how to work in March. And 1st of September, they'll have a 2-week Intergovernmental Conference to discuss what the law should actually contain. And sometime in 2020 or 2030, we might actually see something that can be ratified by different countries. So it's quite a long process to see this happening. And hopefully that will then allow us to have this conservation and sustainable use of deep sea biodiversity. So we've been out to the UN a number of times. I think between us as a group, we had 25 or more meetings with different bodies, from having side events at the UN itself, doing the meetings, to having specific meetings with the African Union, the G77 plus China, the South Americans, the small island developing states, etc. So we tried to fill this between the group of us, and you could see there's myself, Thomas, the other Thomas, Chesty. So this is the conclusions for this first half of the general presentation before I go into the specifics of what we do. I think it's very promising. We see new medicines, new processes, and new materials developing from the marine environment over the last 20 or 30 years, and it's really becoming a powerful force now. I think if done correctly, it will have little environmental impact because we don't go and recover the materials from the marine environment, but that's not necessarily so. When people fish for the fish oils, again, that's a lot of fish have to take for that. The challenges are really still understanding the level of marine biodiversity that's out there and having the proper tools to deal with them. Molecular genetics tools, bioinformatics tools, we don't have proper ones yet. Making sure we have physical access to deep sea, that we have legal clarification of this United Nations Convention laws of the sea. The industry remains interested and we can scale up the materials that we want to the right level. And most importantly, from my perspective, is training researchers with the right mix of skills. So our research center tries to get together people from different disciplines to work on a problem, make sure that we get the best possible outcome. So that leads me into the research center that we have, and our aim is to discover new pharmaceutical candidates and research tools from extreme environments. I say extreme environments, we don't just deal with marine. We started off marine invertebrates, we now work with marine microorganisms from the deep and from the cold, symbionts of marine invertebrates, and also extremely fast from other environments, particularly desert environments. The lab comprises a number of principal investigators. Myself as a chemist, Rainer Abel as a pharmacist, Haideng and Wael Hussein as molecular biologists, Jürgen Eva as environmental chemists, really sort of have the same space, but we often overlap in our research goals, and Laurent Tambo as synthetic chemists. We have a very able spectroscopist, Andrea Raab, and an NMR technician called Russell Gray. What we're really trying to do is to work on the functions of these beautiful molecules, like why they're there for the organism itself, are they involved in symbiosis, how are they made, and how they're involved in binding to inorganic elements. Typically, to pay the bills, we have to do pharmaceutical type work, so cancer, infection, inflammation, parasites, epilepsy, and Alzheimer's disease is what we're working on. So it sounds very broad, So we're focused on molecular diversity, which we can then send out to partners who can test in different areas. The tools that we use typically are also chromatography, spectroscopy, molecular biology, chemical synthesis, molecular modeling, and the development of new software for searching more efficiently. If anybody's interested in marine toxins, that would make that of interest. So the lab itself is an old teaching lab. It's been converted into a very bespoke space for us. So we have in this space here the chemistry space, essentially, which allows us to do the isolation and purification of compounds. A molecular biology lab that's complemented by another microbiology lab in the same building, and a molecular bio lab at the Institute of Medical Sciences. The clean room for working in low metal environments to make metal measurements more straightforward, and a lot of scientific equipment that we use for determining chemical structures, such as big NMR machines. So if anybody's interested in marine toxins, this machine would do very well in terms of looking at very small amounts of substances. If anybody's interested in high-end mass spectrometry, we have two very nice mass spectrometers to determine, what's in a mixture, basically. I like to show this because we do have a very international laboratory. So I think our record is 28 scientists from 18 different countries. It's pretty broad, and it's all very collaborative. So we have basically chemists, molecular biologists, analysts, microbiologists working together, and we often have visitors from different countries. So at the moment we have visitors from Nigeria, Ghana, and Chile in the same lab. So these are people coming in for short stints as sabbaticals or postdocs. And we're very open to collaborations with almost anyone, free of charge to start with, and then as part of a grant later on. So we're interested in extreme environments, like arid desert, which I won't talk about today, the deep sea and cold environments. And I like to work on marine invertebrates when we can still, because it's nice to go collect these lovely things. My colleague works on marine fungi. I like to work on marine bacteria, as is Hydem, and cyanobacteria is Wild-Woosen's area, looking at the biosynthesis of those molecules. And molecular diversity is well, you can't see it, it's very diverse. We try and work on new structural families, so things that are interesting chemically, and get good publications for our scientists in the lab. We're looking for things with new biological activity, but what we have at the moment is from the big European project. We have 5 molecules going into animal trials for epilepsy and Alzheimer's disease. And it's something that we take forward. We'll give you one of those case studies first of all, because it's the furthest. So epilepsy is a disease that's suffered by about 65 million people globally, with about 2.5 million diagnosed each year on top of that. And although there were 25 anti-seizure drugs on the market, for 30% of the patients, those drugs or combinations of drugs don't control the seizures adequately. So we're looking for new treatments. So what we did with this big European project was a process in which we used zebrafish assays for epilepsy screening. So what you do is you take an extract. We had 2,000 extracts roughly. The first assay is basically looking at the zebrafish embryos. If you flash a light at them, they'll start moving towards the light. If you treat them with a compound, before that they don't move towards the light, it's listed as having a neuroactive effect. And about 16%, 70% were shown to be neuroactive at a single concentration. The next one then is you treat your zebrafish larva with a chemical called PTZ, which just gives it a fit. You can see it moving around there. It's easy to track this with the camera. And then if you do it at a single concentration, you test whether or not it reduces the convulsions, and about 30% of the originally neuroactive hits were found to be anticonvulsant. And then you do a dose response assay where you show that if you escalate the dose, it reduces the amount of fitting. And what you get at the end is about 2% of the original extracts were found to be active, and out of those, about 7 had interest in chemistry, out of which really 2 were investigated to the level of animal trials. So again, it's a long process, but again, that was all done in roughly 4 years. So we have 2 patents coming out on anti-epileptics, and the one I show you here were fungal compounds that demonstrated activity in zebrafish larvae and then showed activity in the 6 Hz mouse model, which is what they call the gold standard for drug-resistant epilepsy. Those molecules also had what we would call drug-like profiles, where we tested them to see whether they would go through the gut lining, whether they were bioavailable, whether they got to the target through the blood-brain barrier. They had all those possible effects, which means it's a good molecule to take further. So that's the next stage, is now to try and make enough of this material to take it forward onto the next stage of trials. Let's go and have a look at some examples from deep oceans and cold oceans. So cold oceans with the Norwegians. I worked a lot up in Tromsø and further north, and they were looking at Arctic sea squirts and other invertebrates as well. This particular one here was a useful example. It's my first one to work on, together with Maggie Tedeschi. And she found originally some of these peaks in the HPLC trace They were active against a range of different bacteria. I won't show you what they are, but they all have activity. And what we got out of that was some chemistry that was exciting, mainly for the new chemical structures. So what you see here is the activity is not stellar, but the structures are novel. And they look very similar to something called alezolid, which is an anti-TB drug. It's just been approved, well, one of the most recently approved anti-TB drugs. So we patented it, and the patent was licensed out to a small company called Lytix, who develop anti-infective drugs. So it's interesting to see that you can, even if you get new chemistry that doesn't have great activity, somebody might still want to buy it because you can protect it. It's a very interesting concept. Never seen it done that way around. Typically, they take a molecule that's known, it has high activity, rather than taking a molecule that is unknown, it has high activity. It can really protect the space around it using clever patenting. Example from the deep oceans, and again, I'm sure you're well aware of deep oceans. Again, most of the planet is about 4,000 meters deep. The abyssal plain, or the plain, is not very exciting. But then towards the edges of the plates, you find these deep trenches, such as the Atacama Trench off the coast of Chile. So we try and get materials from those places. So the most recent collection has come from the South Atlantic Trough, just over here somewhere, through one of these Eurofleet expeditions that took place there in December 2016. But this one here, we're able to get materials very early on from Horikoshi, who has since passed away, from the Mariana Trench through their vessel called Taiko, which was lost in 2003. And again, I don't think there's very little capacity to go that deep globally right now. I think the only one that's left is James Cameron's Deep Challenger, which is one of these vessels where he went down into the Mariana Trench and saw what was there. That allowed us to get materials from deep sea. The story is that we got materials from Horikoshi, we gave them to Alan Bull in Kent, who identified new bacteria, or isolated new bacteria, identified by Mike Goodfellow in Newcastle, and gave them to us, and we did the chemistry. Just to show they're properly deep sea bacteria, we managed to put them in high pressure, and at high pressure we can see that they still survive quite nicely. So it shows that they are truly deep sea bacteria. The chemistry out of them is quite novel. So the bottom molecules certainly are novel, I would describe them as, although they look a little bit like clofazimine, which again is an anti-TB antibiotic. But we're interested predominantly because these new molecules have new activity quite often. We had antiparasitic activity shown quite early on, again with Trypanosoma brucei, activity of submicromolar, which is a good starting point for the first one, and no activity against normal human cells, which is the MRC5 cell. So again, it has differential activity. But we decided to look at some of the deep sea work on this. And what we tried is very primitive conditions to put it under pressure. So literally just put it in a food grade bag, put it into your steel bomb down here on the bottom right, and then keep it at the right temperature, and then compress it and grow it. When you do that, if you increase the pressure, you see the profile of compounds produced. This is the normal pressure at the bottom. That's at 10 megapascals, or about 1,000 meters of water. And you can see the profile of chemistry that we know from atmospheric pressure changes. And then you increase the pressure up to 28 megapascals, and still more up to 40 megapascals, which is about 4,000 meters equivalent. You start to see big change in the chemical profile. Now, we haven't been able to reproduce that, but with NERC funding, we're building a piece of equipment which allows us to look at this under what we call polyextremophile conditions. So that means we can take it cold, fairly warm, and also under very high pressure whilst subsampling. So whilst we grow the bacteria, we can subsample every day to test out whether or not things have changed. And no one has been able to do that before. The system is nearly finished. I've set up for about a year now. Hopefully it will be finished actually quite soon. But it will be quite an impressive piece of steel and control system. So it should be quite beautiful. If anybody's interested in working with us on high pressure organisms, we can try, see if it works. So the last thing that we do is development of software tools. Now, quite often there's a big problem that what we find is a lot of known stuff. So this is the same for any environmental work that you do. Quite often you need to know very quickly what's in your mixture. And so we develop tools, and typically it's a very complex process where you take a sample, you do an extraction, you search it in a database. If you don't get a hit in the first database, you check other databases, and it becomes quite an involved process. Most of our students had a long time doing this because it saves them doing a lot of wasteful work later on. So we've developed a number of databases, and the first one we would call targeted dereplication. We'd search every element of the database in your spectrum, rather than serving every peak in your spectrum, searching that into other databases. It seems a bit strange to do it this way around, but it's a good example here of a fungal database. So what we do is we search 5,000 fungal compounds in our spectrum here. We found that most of the compounds were known. We could put a list on it, and then this was the unidentified peak. So we only have to concentrate on that one. It's very simple. That was a new compound. Not a very exciting new compound, but it's fairly quick to find that. So again, I think that's a useful thing to have, and that strategy could be extended to marine toxins or whatever. Another database we have then is all the Streptomyces compounds, so things that we're interested in particular, and it has information about them on there. You can find these compounds then fairly quickly if you have a defined range. So if you look at Streptomyces compounds, you have a Streptomyces, you grow it up, and you want to know whether or not it contains known compounds. This is a really quick way to screen that. This is for us a standard tool. It basically, all the information you have on a compound goes into the database, so that when you search it later on, you put a query here, so it has the structure, it has some basic information, it has the mass spectrum, it has the proton spectrum, the UV spectrum, sometimes a carbon and two-dimensional NMR spectra, so you can really find some things fairly quickly. And although we only started with a small number, that's increasing now because the students are adding their data to the database every day. So you can do a search. There was on the bottom here is a query in green, the hit in red, and they don't look very similar structurally, but the database picked them out, and it turns out they are related compounds in the end. So it was useful for us to use that as a test bed. Finally, a little example from the Ross Sea, from a very cold environment. This is more a story of how you can use the tools to find something, even if the product in the end is not that exciting. What you have is you have your mass spectrum. It searches for peaks. In this, the LCMS trace, and it looks for all the things. So what it does, first of all, it removes the blank. So it removes junk, it removes the medium from the culture conditions, and it removes the database of known compounds. Once it does that, everything that remains must be new, in principle. Okay, so in this case, it's a very tiny peak. It's the only one that's new. So we locate it, we find its match, we find its molecular formula, and eventually it turns out to be this compound family, which again is a known compound family, but they're new analogs. So again, it did work. We published a strategy in Marine Drugs, which is a very specialist journal of this area. But I think it's a good example of showing you how you could use these databases to find things. And again, this could be applied to any environmental research that you choose to pick. So in the end, I'd like to conclude. I think we do get a lot of new biodiversity from marine environments. We get new and novel bioactive compounds. New being derivatives of known compounds, novel being things that have unprecedented chemical structures. And those are the ones that we get excited about as chemists. I think we can manipulate the culture conditions of any of these things to get new profiles. So we see that quite often. So if you increase the pressure, we see a change. If you increase the culture conditions, you see a change. I think we can also get a sustainable supply of materials using this. Again, we don't have to worry too much. We can grow the bacteria under specialist conditions or manipulate the genes to produce the products that we want. I think these tools at the end do streamline the process. It means that we find something, we can whittle down thousands of bacteria down to a few fairly quickly using biological testing and chemical screening processes as well as genetics. So I'd like to thank all the people that did the work, of course. I haven't written down everybody from the Pharmaceutical consortium because they wouldn't fit on. There's 60 of them. There's a big European program that's finished last March. But I would like to thank predominantly the principal investigators in each one. So Margit suggested the work on Synoxazolidinones, while did the work on the deep sea magazines, and Joji worked predominantly on the tools for dereplication. And finally, people who paid the bills. There's quite a few agencies. Typically, the work that I described today, a lot of it was done by Pharmaceutical consortium. Some of it was paid for by the Norwegian Research Council and the Egyptian Research Council. So thank you very much. Hopefully you have some questions.